Dear students, today our topic is about the meiosis. Meiosis is a specialized type of the cell division that occur in sexually reproducing organisms to produce the gametes, such as the sperms and the egg. We can define the meiosis is basically the formation of the gametes, such as the egg and the sperm. It's also called the reduction and the division process. It proceeded by the interphase which include the chromosome replication. So the meiosis have been divided into two mitotic division, the meiosis first and the meiosis second. Here the original cell is diploid, which is the 2n number, and all the four daughter cells produce that are the haploid number, which is the n number of the chromosome. So the significance or the importance of the meiosis is that here the two haploid n number of gametes are brought together through the fertilization to form the diploid 2 number of zygote. So the primary purpose of the meiosis is to reduce the chromosome number by half so that when the two gametes fuse during the fertilization, the resulting zygote restore the original diploid number of the chromosome. So what are the importance of the meiosis? Say that meiosis must reduce the chromosome number by half, and fertilization then reduce the 2 number of the chromosome. It's mean creating the genetic diversity through the processes of crossing over and independent assortments of the chromosomes occur. So in the meiosis first, which is the prophase first, is the sequence of the phases subdivided into the periods known as the Leptotena, Zygotena, Pchytena, Diplotena, and Diakinasis. So it's mean that prophase first is the longest and the most complex stage of the meiosis, and characterized by pairing of the homologous chromosomes and genetic recombinations, which we call the crossing over. So the first step in the prophase first is the leptotene, a thin thread-like structure formation. Here the chromosome began to condense and became visible as long, thin threads-like structure. So the importance of the leptotene stage is it marks the initiation of the chromosome condensation. So here the picture showing that the leptotene is the thin thread-like structure. The second part of the prophase first is a zygotene, which is the pairing of the thread-like structure. The picture showing that the synapses began, for example, homologous chromosomes start to pair up and align along their entire length. A protein structure called the synaptonomal complex forms between them. For example, a protein complex forms between these two threads, and the pair structure is called as a bivalent and tetrad for chromatids. So what are the significance of the zygotenes, that is the pair threads? It crucial for the precise alignments of the homologous chromosomes, which is necessary for the chromosomal or crossing over during the homologous combinations. The third step in the prophase first in the diplotene, which is the double of the threads. Here the picture also showing that the synaptonomal complex dissolves and homologous chromosomes begin to separate from each other. Now they remain attached at the sites where the crossing over occur, which are now visible as X-shaped structures called the chiasmata, which is the singular one is the chiasma. It's mean that the double threads like structure arrange them in such a form that it looks like the chromosomes. So what is the significance of this event? Basically, the chiasmata hold the homologous chromosomes together, ensuring their correct segregation. For example, during the homologous recombination, all the events correctly segregate toward the next event. The fourth one is the diakinasis. Diakinasis is the moving apart of the event. Basically, it is the terminalization of the chiasmata occurs. The chiasmata move toward the end of the chromosomes. Chromosomes are fully condensed. So the nucleolus disappear here and all the nuclear envelopes break down. So what is the significance of the diakinasis? It's prepare the chromosomes for separations during the metaphase first and the anaphase. So these are the summary of all the four different events which occur in the prophase first. After the prophase first, now the second phase is the metaphase first. The picture showing that here the homologous pair of chromosome align along the equators of the cell. Basically, the pair homologous chromosome here we call them as a tetrads align randomly along the cell equator, which is the metaphase plate. This independent assortments contribute significantly to the genetic variation. This one is the event where the genetic variations can be occur. Anaphase first. Now in the anaphase first, all the homologous separates and move to the opposite poles. For example, the picture showing that the sister chromatids remain attached at the centromere. The homologous chromosomes separated and pulled to the opposite poles of the cell. So the sister chromatids here remain attached to their centromere. Telophase first. After the anaphase, here the telophase picture showing that the nuclear envelope become reassembles. They start to begin again in their shape. All the spindles fibers which we call the microtubules are disappeared, and now the cytokinesis divide cell into two. For example, the cell is ready to for the division. The chromosomes arrive at the poles, the nuclear envelope may reform and the cell divides, which we call the cytokinesis, resulting into two haploid daughter cells. Each chromosome still consists of the two chromatids. Now the second phase have been start, which we call the meiosis second, which start again with the prophase second. Now in that case, the cell which was separated start the prophase second. Here again the nuclear envelope fragmented and the spindle microtubules formation have been occurred. So the meiosis second, which is the equationally division, this division separated the sister chromatids and is similar to the mitosis. There is no DNA replication between the meiosis and the meiosis second. Prophase second, which is the chromosomes condense again if the disc condense in the telophase first. So the nuclear envelope break down and the spindles apparatus reforms. So in the meiosis second, which is the metaphase second, here all the chromosomes align along the equator of the cell. So the metaphase second, which is the chromosomes each with two daughter sister chromatids align individually along the cell equators. In the metaphase second, which is called the anaphase second, here all the sister chromatids separated and move to the opposite poles. Here picture showing that the sister chromatids separate now the considers individual chromosomes and are pulled to the opposite poles. Meiosis second, in the telophase second, all the nuclear envelope again assembles, the chromosomes again discondense, all the spindles fibers will be disappear and the cytokinesis divide the cell into the two. So the telophase second and the cytokinase second, the separated chromosomes arrive at the poles, nuclear envelopes reform and the two cells divide, resulting in a total of four non-identical haploids daughter cells gametes. Here the picture showing the result for haploid cells with one copy of the each chromosome. So this one is the summary of all the meiosis first event, which start with the early prophase first, which is subdivided into four subdivision, then the late prophase, metaphase, anaphase, telophase, and the cytokinasis. Again, the summary of the meiosis second have been shown, which have been subdivided into the four identical haploid cells. So this we can differentiate between the two events, which is the mitosis and the meiosis. So in the mitosis the number of division was 1, while in the meiosis the number of division was 2. In the mitosis the number of the daughter cells was 2, while in the meiosis the number of the daughter cell were 4. In the mitosis, they're genetically identical, while the meiosis, there is no genetically identical events. In the mitosis, the chromosomes are same as a parent, while in the meiosis, the chromosomes are half of the parent. The mitosis are mostly occur in the somatic cell, while the meiosis mostly occur in the germ cell. The mitosis are occur throughout the life, while the meiosis occur only in the sexual maturity. Lastly, the mitosis play a efficient role in the growth and the repair mechanism, while the meiosis playing during a sexual reproduction, childs have been born. So these are all about the meiosis first and the meiosis second step of the cell division. Thank you so much.